Olive or Olea europaea is a very important species for anybody who lives around the Mediterranean Sea. It's a diploid organism with 23 pair of chromosomes, mostly allogamous. The origin and the history of domestication of olive is very, we say it's lost in time, because a lot of people came up with hypotheses based on different studies, but it was mostly phenotyping, using the shape of the fruits or the shape of the leaves, of the colors, but nobody could give us if you want, a snapshot of the history of olives. But recently, with the genomic era, with the utilization of sequencing and the bioinformatics, we started getting the dust off of the origin of olive trees. So based on transcriptomics and based on transposons, they said that the olive domestication started in the Levant, which is Syria, Lebanon, Palestine, and it moved west toward the Mediterranean countries, north and south of the Mediterranean, because of the migration movements. So now we have an idea where the olive came from, but now we have to talk about the diversity of olive trees. If you look at all the different varieties and cultivars of olive trees, they look all the same. Sometimes even the fruit looks the same, the leaves looks the same, but there is a fraction of this, what we call a phenotypic plasticity, that could not be explained by what we have now. So we have to dig deeper into the DNA or the RNA, or sometimes into the cis element and the regulatory element of those genes to see the actual fraction. What is that variation in those little tiny pieces of DNA, if they can or not allow us to explain that fraction that we could not explain before. So what we did first, why is olive oil better than any other oils? The question is, the answer is the concentration of the oleic acid, which is the most important fatty acid in the oils. The oleic oil is what given the olive oil the taste and all the properties that we love it for. If you compare it to sesame or the soybean or even the argan oil, the content of oleic acid is way higher. Sometimes it gets to 83%, which is what all the other industries are trying to do. If you look, for example, for the soybean industry, especially here in the United States, is trying to push that oleic acid content from 50% to a 70 or 75 or 80% to be competitive with olive oil. But here the story, why is the oleic acid content in olive oil higher than every other species? We're going to unfold the story and we're going to use the genomic to explain that difference. So the first thing we did, we sequenced the ancestor of what we are today calling an olive tree, which is Olea europaea var silvestris, which is the wild olive. Sometimes we call it in Morocco jbouj. It's a little bush. It looks like an olive tree, but it's smaller. The fruit are smaller. The leaves are smaller, but the quality of that oil is better. We're gonna understand why. So we did a shotgun sequencing. The shotgun sequencing of a genome, when you take a whole DNA and you do a sonication, you shear it, you bombard it. Shotgun. Shotgun means le fusil à pompe. So we hit a bullet in that genome and we cut it in billions of pieces, and then we run it through sequences. So we sequence it on Illumina HiSeq 2000 on parent libraries, So when you have those little fragments, we sequence them both ways, forward and reverse. On a 220x coverage. A 220x coverage means that every base was read 220 times. The higher the coverage is, it's easier for you to assemble the genome and you know that your data is adequate. So the sequencing resulted in 515 gigabytes of raw data. And after the assembly, we assessed the draft assembly size of 1.48 gigabyte base pair, which is the size of the olive genome. That 1.48 gigabase pair was confirmed by a wet lab analysis when we do cytometry en flux. We estimated the genome in silico using algorithms and using wet lab, and it came to the same thing. So the thing very interesting about the olive genome is it 51% composed of repetitive DNA. And a lot of people, they think that repetitive DNA is what we call junk DNA or trash DNA, but it's telling you a lot about the evolution of that species. But here, if we compare the wild olive that we sequenced to the cultivated olive, the draft genome is 63% repetitive DNA. And this is a big faculty of plants. And Abdulmajid will correct me if I'm wrong. Plants, they tend to have a lot of repetitive DNA in their genomes because of the hundreds of thousands of years, if not millions of years of evolution with duplications and rearrangement, and those repetitive DNA, they just get compact into the genomes. So when we sequence, we assemble the genome, the next step is to annotate the genome. The annotation is take a part of that DNA and tell me what is it. Is it a gene? Is it a transposable element? Is it a cis regulatory element? So we adopted an approach using three methods. We start with de novo annotation, and that's using a heavy algorithm like Augustus, and it's using open reading frames. Open reading frames are a sequence of DNAs that will tell you where the gene is starting. It's an open reading frame. That's when you start. The DNA polymerase go through that and start reading your DNA. That's the first approach. The second approach, we used a similarity approach. The similarity approach is when you take a sequence from a new organism that you just sequenced and you blast it. A blast means a basic local alignment. You align that sequence to a database, and that database had genes and organisms already annotated with all the information we need about them. The third approach is we sequence the RNA of the wild olive tree. When you sequence the RNA, what you have is the expression, is the messenger RNAs. It's what giving you coding genes. So we have those three the approach with three different methodologies. When we combine them together, we've been able to predict over 50,000 protein coding genes. And then out of those 50,000, we anchored. Anchored means that we've been able to find the exact position of those genes in the chromosomes. And if you look at the picture on the right, the bottom one on the right, that's the number of your chromosome. That's 23 chromosomes. And those different shades of colors, those are talking about the density of the genes. For example, a dark color means that there is more genes in that area. A lighter color will be telling you that there is less genes in that area. So after sequencing, assembly, annotation, now we're going to start a downstream analysis to tell us stories about that genome and its evolution. What you're looking here right now, this is my favorite diagram. It's called the Manhattan diagram, which is basically the genomic landscape of the wild olive genome. What you see there, it's everything that exists in that genome with all the genetic features. We're going to go from outer and we're gonna go all the way to the middle of the circle, and I'm gonna explain every feature, what's in there. So the first circle that we see, let me just use my technology. So here, in this layer right here, these are the chromosomes. You see different sizes, different colors. These are the 23 chromosomes in the white olive genome. We go a layer in, and it's telling us about the genes. The genes, as we talked about right here, I don't know if you see, can you see it?
Yes.
Can you see when I modify here?
Yes, we can see.
Yes, yes.
So these are the genes. I'm gonna try to zoom in right here. Oh, no, let's go back. It's not allowing me. So here again, these are the genes in every chromosome. This is just a manifestation of all the data. We're talking about a lot of data, but we put everything compact in a beautiful visualization like this. So the layer A1 is all the genes that are in that chromosome. Something that you can tell just from looking at this, you can tell which chromosome has a higher density of genes or had more genes. If, for example, we make a comparison between chromosome 18 and chromosome 12, we can see that the colors are dense and compact in chromosome 18, but there is a little white stripes on chromosome 12, which means that there is more genes in chromosome 18 than 12. So this is just to have an idea. We go to the layer A2. The layer A2 is the messenger RNA. So for every gene, you have a coding part and you have a non-coding part. You have exons and you have introns. So which makes the whole messenger RNA before splicing. A3 are the ribosomal RNA. And you know what l'ARN ribosomal does is helps in the translation. You have the 2 units, they take the messenger RNA, that's translation to give you a protein. The A4 in yellow, right here, is tRNA or transfer RNA. And these ARN de transfert, they help into moving compounds and helping in post-transcription regulation. In B, that's the gene density that we used to talk about. And it's the darker the color, the higher the density is. If you look at, for example, chromosome 19, really small chromosome, but really dense in terms of gene. RPKM stands for read per kilomillion. And this is how you can assess the gene expression. A gene, when you express a gene, the number of reads, when you sequence that RNA, the number of reads will give you an idea about if that gene upregulated, is it downregulated? It depends on the condition. For example, you are taking, as you were talking about bacteria, you take that bacteria in a Petri dish with the concentration of this kind of antibiotic, and you use a different one, and then you sequence both of the RNAs, and it will tell you what are the genes that are actually involved in the resistance to that, based on the gene expression of that bacteria. D is the tandem repeats. Tandem repeats are repetitive chunks of DNA that are repeated in tandems. And then we have the coverage of transposable elements. On F, transposable elements are very interesting because they're not coding genes, but they regulate the expression of other genes. We're gonna have an example, we're gonna talk about it. The last one in G, which is the interchromosomal symphony, right here, what you see right here in the middle, And this is giving you an idea about the duplications events that happen in that genome. So when you see a color coming from here and go all the way to here, which means that that block from this chromosome duplicated and went to the other chromosome. So all those sentinel layers giving you ideas about the duplications in the genomes. So here is the very important or very interesting part is when you're talking about the genome evolution and the duplication events. Very important. So in the history of olive, we had two major duplications. An old one 60 million years ago that was estimated by 4D-TV. If you are interested, I can, at the end of the presentation, we can talk about the 4D-TV analysis and how they estimate time of divergence, of duplications, of speciations event. And there was a recent duplications around 25, 30 million years ago. If we look at the first one, 60 million years ago, it's almost coincide with when the biggest chunk of outer space hit Earth, and we almost lost 3/4 of the diversity. So if you think about it, the olive genome, to survive those conditions, had to duplicate its own genome to survive. So which makes sense when we study the speciation event. If you look at the right here in the middle, this is telling you about the speciation event and duplication event, and this is very important. I'm going to explain to you what's going on. Sometimes you have two species, like right there. We are talking about sesame, le sésame, and an olive. 71 million years ago, those two species were one species, and a speciation event happened and separated the two species, olive and sesame. And after that, you had the duplications event. That's why when we want to study the olive, we compare it to sesame because they came from the same ancestor. And we're going to explain this more. In the third sphere right here, it's a Symphony Analysis. You can see it right here, chromosome 22, for example. This chunk right here, if you go and you take it from the genome and you compare it to this chunk in chromosome 4, they're going to be identical in terms of blocks. I'm not talking in terms of sequence, but in terms of blocks, because with the duplication, there is mutations that can change a little bit, but the block will be the same, which means the disposition of the genes. If there is any, Moulia Abdelmajid will correct me, if there is any linkage group that we use in the QTL mapping. And this is very important when you have blocks like that. We were talking about the duplication events and how important are they to tell us the story about genomes. This is what happened. When you have duplication of genomes, you have duplications of genes. And when you have duplications of genes, we have a copy number variation. The expression of those genes is higher because there is more copies. So what happened, we took the sesame, le sésame, and the olive. They were the same species, we said, and we had a speciation event, and then we had duplications. Look what happened. In the oil biosynthesis pathway, which is the most important pathway in olives, in sesame, in soybean, in argan, because that's where the money is. Look here what happened in the SACPD. SACPD genes are genes that transfer the stearic acid into the oleic acid, which is very important. So here in sesame, we have only 2 genes, but in olive, we have 7 genes. So what happened is those 2 duplications, they took the number of genes from 2 to 7. But someone will say 2 times 2 is 4, and then 2 times 4 is 8. We should have 8 because we have 2 duplications. But what's going to happen is sometimes the genome will kick away 1 copy of those genes. It doesn't have to keep all of them. It depends on the rearrangement or what the genome needs at that time. So SACPDs, we have 7 copies. A very important gene in the family, in the oil biosynthesis pathway, are the fat family, the fatty acid desaturase. So what happens when you have the stearic acid, the SucPD will turn that stearic acid into a oleic acid. And then the FAD2 family will take the oleic acid and transform it into a linoleic acid. Here again, 2 versus 5. We see that the duplication event, they expanded the number of those genes. But this is very important. If you're not familiar with the oil biosynthesis pathway, I'm going to explain to you how it works, because the rest of the presentation that was the amazing thing about the research that we did and the publication, because when you have 7 SucPDs, just think about what we talked about earlier. There is more oleic acid in olive than any other species. Why? Now we're going to explain why. Imagine if you have 7 members of your family bringing food to the table. So you have accumulation of oleic acid, but then you have a lot of people coming to eat, which is the FAD2. So if we are pouring food from the SucPDs, more oleic acid, but the FAD2s are taking the oleic acid and transforming into linoleic acid. But still, the concentration of oleic acid is still higher than any other species. Why? If we have 5 FAD2s taking all that oleic into linoleic, we should have less oleic, not more. But zaytunatun mubarakatun is in the Quran for a reason. So why? Here is why. So 5 of those genes, only one of them is working, because the other 4, this one, this one, this one, and this one, there is a siRNA, means a small interferon RNA derived from a rich region of transposable elements that we were talking about and how they regulate the gene expression. So what happened is we have more oleic coming, but the genes that are going to transform that oleic into linoleic, they're not working because their expression is stopped by this small interferon RNA right here. So we have only one FAD2, which is 3 in the chromosome 17. This is the only one working. So what's going on? There is no desaturation of oleic acid into linoleic acid. And that's why when we published this manuscript, all the studies just boomed. If you look at any studies before 2017 on olive, they were very, very limited. As soon as we published the chromosome level sequence and we did the evolution of oil biosynthesis, if you look now, there is studies all over if you go to ResearchGate, just put olive genome and you can have a huge amount of studies coming, because of now we know what's going on in there. So this manuscript was published in the Proceedings of National Academy of Science in 2017, which is the 2nd most cited journal in the world. This is what basically allowed us to publish there, because this is very important. Okay, so this is the story of the genome. This is the story of what's going on in terms of genes, their regulation. Now we're going to go to a different kind of approach. Here we studied genome, that's genomics. We studied transcriptomics because we sequenced the RNA, and then proteomics because we're dealing with proteins. So now we're going to go to a visualization. How can we look at all this? How can I go and look at that position and see what's going on in there? I need a genome browser, just like internet. You want to look for something, you go internet and you put a request, and all the databases and everything, how it works, and then they give you the answer. A genome browser is the same thing for genomes. You want to see genes, you want to look at where they are, their positions, what they do, are they involved in any networks? You go to what we call a genome browser, and this is what we're going to talk about right now. So the Olive Genome Browser was made on a CentOS. It's an operating system that deals with MySQL databases. It seems very complicated, but it's very straightforward and very easy. So basically what we did, we made a database based on a genome flat file. What is it? Is you take coordinate, les adresses of every single feature that we talked about, the genes, the messenger RNAs, the tandem repeats. You take their coordinates from the genome, you put them in a flat file, you build your database, and you feed it to the gbrowser, which is the graphical user interface where you can see this. All we see right here is to give you this at the end. So we're gonna go step by step to look at this genome browser. So here we're looking at chromosome 2. Right here, chromosome 2. We're looking at this specific area right here, which is a 200 kb. And it's giving you all the details of genomic features. You wanna see if there is any transposable elements in the region that you're interested in. Let's say that's a genome of a bacteria. And I wanna design primers to amplify a certain gene. Yeah. I need to go here, for example, you gotta go to the messenger RNA, and you look at this area right here, and you can zoom in, and it will give you the sequence. It's zoomed down to the sequence of that area. And if you know that area, you can design your primer for something to overlap with that, so you can amplify the genes that you want, for example. Let's say I'm working on genes that are involved in a polyphenol biosynthesis. I can go there with the gene ID, I can put it right there, and it will give me all the information, the positions, everything, the splicing sites, if there is anything that's controlling that expression. So genome browser is very important. So this is the story of the Olive genome. Now we're gonna go to a very this subject is dear to my heart because I had to fight the consortium so we can dig in this area, because everybody thought that, okay, we found the FAD2 gene family, it's controlled by small interfering RNAs, it's epigenetic, it's like like we say back home. But I told them maybe there is a different kind of regulation still in the oil biosynthesis pathway. So the new fashion nowadays in plants is to study the DNA methylation. DNA methylation is an easy switch for gene expression. It's just like a switch on and off for the light. You have methylation, that's a switch off. Methylation stops the gene expression, because methylation is you have a cytosine that will be methylated, and a methyltransferase will put a methyl, CH3. It will put a methyl on that C base, and it blocks. It blocks the expression, because any transcription factor that will bind to that area cannot bind because there is a methylation, so it stops the expression. So this is how we started. I was like, maybe we can look at the DNA methylation and see if we can find any different kind of regulation. And this is what we did. So how can we, in 5,000 years, or probably more, how can we go from this to this? Can you see the circles on the pictures?
Yes.
So we have on the left, it's a wild olive tree. And on the right is a domesticated, it's a cultivar. And you can tell. But imagine if you have thousands of those cultivars, how are you gonna separate them? So the studies started with RNA. They took the wild type, they took the cultivated type, they took wild type from the Eastern Mediterranean, from the Western Mediterranean. They took cultivated from the Eastern Mediterranean, the Western Mediterranean. They couldn't see any difference in the genes. So they concluded that the 5,000 years of domestication of olive didn't affect the variants, didn't affect the genes. Another study came, that one was in 2017. A study came in 2019 and said, in 2020 and said, well, we can link that diversity that we see, that plasticity in phenotypes, to the activation of transposable elements ahead of genes. And that's why it's controlling the expression. I'll give you a simple example. We have two patients. They are both screened for BRCA1 breast cancer genes. One of them got cancer, the other one doesn't have cancer. How come? We have the same genes. You have a regulation of that gene. One is operating, it's working, the other one is not. And this is what DNA methylation does. It switch on the expression or switch it off. So we're trying to explain via DNA methylation the difference that you are looking at on those two. Because if you go to the genomic sequence, those two are very, very similar to a very confusing point. So we're gonna dig deeper and get away from the genes and study the regulatory elements of the genes. So to do so, we opted for a profiling using MedipSeq. MedipSeq is a technology that uses antibodies to immunoprecipitate the methylated DNA. So how it works, we took DNAs from the leaves, we took DNAs from the fruit. The first step you do is the sonication. You have a big piece of DNA, you want to cut it in little pieces so it's easy to process. So we take it, we cut it in pieces by sonication. It's c'est une sonde, and it will break that DNA into little pieces. When you break the DNA into little pieces, you have to denaturate the DNA so the two strands separates from each other. And then when we do that, we have these chunks right here. These chunks right here, if they are methylated, we use an antibody called anti-5-MC. 5-MC stands for methylcytosine. The fifth base is methylated. So basically what this antibody does, it's right here, it can recognize anything that is carrying a CH3 on the C base. So it will bind to this one and everything that is methylated. After that, we use protein G dynabeads, and those beads, they are magnets, and they can bind to the other side of the antibody. So basically what happened, the antibody will recognize the methylated DNA on this side, but on this side can bind to those beads, the beads right here. Why we use this technology? Because later we want to separate the methylated DNA from the unmethylated DNA. And the magnet will grab all of those DNA beads, and we know that those DNA beads are ligand to the antibody, which is hooked to the methylated DNA. And then when we're done, we do a purification. Those beads, they will stick to the l'épandard, the tube. It will stick. Yeah. We'll clean our DNA, get rid of whatever it's not methylated, and all we have in there is methylated DNA. So this gonna go into the sequencer right here for sequencing. Why we want to do that? Because we want to know what sequence is methylated, so we can know where is that sequence and what is the function of that sequence. So when we put them in a Illumina HiSeq 2000, that's what we use. It will produce what we call a FASTQ file. A FASTQ file is right here of a 49 base. So the reads that we had, they're small. That's the only problem with this technology. The reads are very small. And the problem when you have a small read, when you want to align it to the reference genome to know what exactly that sequence is, those sequences because of what we talked about, the repetitive DNA, they can bind to different areas. And that's a problem. So when we have that sequence, that usually comes out of the sequencer, or if you don't have a sequencer, you can ask a company to do the sequencing and they will send you what we call a FASTQ file for every sample. So once we have that FASTQ file, we're ready for our downstream analysis. So the downstream analysis, this is how the pipeline works. We start with a FASTQ data pre-processing. A FASTQ data pre-processing is we take those sequences, we want to know if they are good quality or not. So we do a fast quality control. The fast quality control will give you an idea about the score of your sequence. Are they good or not? Where the score is really good, where the score is bad. And then we do a trimming. You trim the adapters that we use for the sequencing, and you trim all the reads with a lower quality than the threshold that you set. When you have clean data, we go to the second step, which is the mapping against the reference genome. So what is the mapping against reference genome? You have a set of reads, and you're looking at the overlapping between your set of reads and the reference genome, and that's the mapping. When you map, you get a BAM file. You take that BAM file, you run some statistics on it, so you can know. Sometimes 3 sequences will map to the same area, or the same sequence will map to different areas. So what the algorithm will do is calculate the p-value. The highest score, the sequence with the highest score will be allowed to map in that area. So when you have your BAM files, you are ready to call those peaks. What does it mean? Those sequences, when they map to a spot in the genome, it means that there is methylation in there. And we want to assess the signal of that methylation. So we do a peak calling. And peak calling can be done with a lot of algorithms. There is a lot of tools out there. We use Max. Max is a very straightforward tool, and it pays attention to methylation that is on non-CpG islands. Anybody who's working on human epigenomics, they concentrate on CpG islands because that's where the majority of the methylation is. In plants, it's different. There is areas where there is no CpG islands. CpG islands, just to give you an idea, is just a lot of C's and G's, a lot of C's and G's, and G's and C's and C's and G's. And this is CpG island. So when we call those peaks, we know, if we look at it right here, this is a gene right here. This is a gene, and these are all signals of different methylations. And it's very important to know that, especially when you have methylation in the transcription, in the promoter region. The promoter region is usually between 1 to 2 kbs upstream the gene that has all the cis regulatory element that control the expression of this gene. So maybe here, for example, all this area, there is a transcription binding site that allows the expression of this gene, but because there is a methylation right here, it's not gonna allow the binding, and then it's not gonna have expression for it. It's gonna be silenced. This gene gonna be silenced. And that's the case of a lot of cancers. That's how they work. So after the call of the peaks, so we know where actually is the methylation in genome, we have to annotate that. We need to know the exact same position, and we need to know what is the genomic feature. Is it in a transcription starting site? Is that methylation in a transcription termination site? Is that methylation in an exon or an intron? Different genomic features have different meanings. For example, if you have a methylation in the promoter region, it's most likely to control the expression of that gene. But if you have a methylation in the intron, it's important, but not as important as in the regulatory element part, which is the promoter region. And we're going to get to this later. So this algorithm, what it does, it will find you the position of the methylation and look around it. And see if there is any gene around that position. and it will give you a score. For example, right here, I'm going to try to see it right here. That TTC means transcription termination site. This number, if it's a positive value, it's going to be behind the genomic features, behind the TTC. If you have, for example, let's find something with a TSS, which is right here. TSS means transcription starting site. And that's the most important type of methylation because that's where the transcription of the gene starts. And if you look here, you have minus 982, which means that your methylation is 982 bases upstream. The negative value means upstream your transcription starting site. And that's very important because you need to investigate if that methylation is actually causing a silencing of your gene. After the annotation, so you take all your methylated genes, especially if you have studying two conditions like we're doing right here. We're studying the methylation status in the beginning of the development of the fruit and at the ripening of the olive fruit. So what we're looking for is differential methylation between when the fruit is starting the development and when it's ready. And we also compared the wild olive with the cultivated olive to see the domestication fingerprint on the DNA. We also compared the fruit to the leaves. The fruit is very important. The leaf is very important for different reasons, but we just try to compare them because in the literature, there was no previous DNA methylation comparison between organs. It's always the leaves or the roots, but they never compared fruits and un organe végétatif, un organe reproducteur. That's the neos that we're trying to do. So when you find all those genes, you want to know what's the functions of those genes. Okay, yes. I have this gene, it's methylated, then what? I need to assign a function to that gene. That's what we do enrichment analysis. We do gene ontology analysis. Gene ontology analysis are usually, they give you an idea about, if we look right here, they will give you an idea about the biological process. The biological process could be a replication. This is a biological process. Don't you see if they will tell you your gene has gene ontology related to RNA transport or DNA polymerase or replication or something. It will give you also an idea about the molecular function. If you have an enzyme, for example, is it a reductase, isomerase? It will give you even the enzyme code to that one. And it will give you an idea where actually your gene is evolving, is working. That's the cellular component. Is it on the membrane? Is it in the thylakoid? Is it in the appareil de Golgi? It will give you where exactly your gene is working. After the gene ontology, we do what we call a KEGG pathway analysis, and this is very important because now we're going from here to deep down into the pathway. The KEGG analysis will give you an idea about where your gene is exactly involved. For example, here we're going to see linoleic acid metabolism. So if my gene is differentially methylated and it's involved in the linoleic acid metabolism, I need to know what is it doing in that linoleic acid metabolism. And we're going to look at all of this further down the presentation. We're done with our genes. We've assigned functions to them. But as we said, even comparing the wild and the cultivated, they couldn't find any differences when it comes to the gene structure or even their expression. So maybe we need to look at the regulation of those genes to be able to explain the differences. And that's what we did, the Cis Element Analysis. Cis Element Analysis, you take a 1 kb or 2 kb ahead of the gene. The gene is right here, and you take this area, which is the promoter, and that's where actually all the regulation is going on. La machinerie de la régulation, that's where it happened. So we took all of that promoter region, and we did cis elements. Cis elements means all of these little things that you see, these different little colors, these are transcription factors. These are all these cis elements. They control basically a lot of gene expression. So now we're gonna go to the results of what we did, and I'm gonna go point by point and explain every step right here. So we took our DNA, we took the FASTQ file, we mapped. After the mapping, we called the peaks, and we want to do an assessment of the methylation. There is two ways to do an assessment of methylation, either by the number of the CpG islands or by the differential methylated regions. So we have genome A, which is wild, genome B, and we go to certain regions, and if there is a methylation on this genome, but there is no methylation on this, those are differentially methylated regions, and they will give you an idea about what's going on. So here, we took the wild, we took the cultivated, and we just compare the methylation in the leaves and the fruit. This is the first thing that I told you about. We compared two different organs. Vegetative organ, which is the leaf, and un organoproducteur, which is the fruit. We found out that there is more. If you can see it right here, the orange is the leaf samples, and the greens are the fruit samples. We have different genomic features. We're starting at the transcription starting site, transcription termination site, introns, exons, and the intergenic regions. So why do we have more methylation in the leaves than in the fruit? So to answer this, we looked at the gene expression of the methyltransferase. The methyltransferase are the enzymes or the genes that methylate the DNA. So we took the methyltransferases from the leaves, and we took the methyltransferases from the fruit, and we compared the expression. So if you see here, this is the sample, this is the heat map from the leaves. If you look at the colors right here, they're pretty dense. And to compare them with the fruit, so here we have more expression here. So what's going on is in the leaves, the methyltransferase work more than in the fruit. And that's why we have more methylation in the leaves compared to the fruit. And we were looking in the literature, and there's no comparison. Maybe people are not interested in this question, but we just wanted to do it because it wasn't done before. And maybe it will bring some insight and people will be more interested in this. So now we compare the leaves and the fruit. We know that there is more methylation in the leaves than in fruit. And if you want to think about it, push it a little bit further. In the leaves, the leaves very complicated organs. La photosynthèse, there's a lot of things going on. And there is also a lot of what we call housekeeping genes. Housekeeping genes, they do the little jobs. They just maintain the DNA and all this. And you have to switch them on when you need them, and you have to switch them off when you don't need them. And the easiest way to do it for a genome is by methylation, on and off. Methylated, stop it, and methylated, and it will work. After that, we compared actually the leaves and the fruit. We didn't compare them, but we compared within the leaves and the fruit samples to see if the methylation has anything to do with the ripening of the fruit. The question why, because there was 2 or 3 studies in 2 different kinds of fruits. There is 2 kinds of fruits. There is climateric fruits and there is non-climateric fruits. A climateric fruit like an apple, when you harvest the apple and you put it on a table, it will continue to maturate and ripen because it's using ethanol, alcohol to maturate. An olive, it's a non-climateric fruit. When you harvest an olive, it's gonna stay just like that because it's using the abscisic acid to maturate. So it's an apple, if you leave it, you're gonna smell that alcohol smell to it. That's the ethanol ripening the fruit, even if it's not on the tree. So that's why we wanted to answer this, because they say that climateric fruits, they have a hypomethylation during the ripening. Non-climateric fruit like orange, they have a hypermethylation with the ripening. So pour trancher, if the type of the fruit is related to the status of methylation, we compared. Surprisingly, in olive, even being a non-climateric fruit, we saw a hypomethylation during the fruit ripening, which is against everything that we've been told, but there is just few studies. And then a study on strawberry, another non-climateric fruit that had just been published. It's actually, they said, even if it's non-climateric fruit, we are observing hypomethylation. Here's now another answer, which means that the type of fruit has nothing to do with the state of methylation during the ripening. And this is a very important for l'arboriculture, because there are sessile plants. Methylation is very important because that's the only way those trees can adapt. Give you an example. An ash tree that lives in Scandinavian countries, 3,000, 4,000 years old, in the same spot, the only way that tree can adapt is by methylation. Because it's not such an espèce. It's going to be there for hundreds of thousands of years. It's not like other species. You're going to grow them this year and harvest them. Next year, the conditions are different. They can adapt. And then you get another species, but the tree, it's not moving anywhere. It's not going anywhere, and needs a mechanism to adapt. And that's why people are now getting interested in studying DNA methylation landscape in response to climate change. So here we compare, as I said, we compare the fruit in July, still like a little olive green, and we compare it to a ripe olive, which is black or purple. So what happened is the methylation stops the photosynthesis in the fruit. And the first step, the green olive will turn a little brownish yellow. That's the degradation of chlorophyll into carotene. That's the first step. And then the yellow or brown olive will turn purple. That's the degradation of carotene into anthocyanin. And that's the role of DNA methylation in the ripening of the olive fruit. It's controlling all of that, plus the abscisic acid and all different cascade of genes. What is going on, it's turning off the light on a lot of things for that olive to go from green to black. But we observed a different tendon in the leaves. So in the fruit, for maturation, the methylation will drop, but it will go up in the leaves. If you ask yourself why, because we compared here again, we compared the methyltransferases in the leaves between July and November, and we see.
Usama, sorry to interrupt you. If you can wrap it up in 5 minutes, so to give a little time for Q&A. Thank you.
All right. I'm going to go to the very important part then. Right here, 2-3 minutes. So the most important genes that we were looking at are the genes involved in the polyphenol. If you take a Moroccan olive oil and you try it, it will give you a little burnt right here, a little burnt sensation. That's because of the polyphenol. So if you compare a wild olive oil and you compare it to a cultivated olive oil, this one is bitter. Une sensation de Why? Because of the polyphenol. So we found out that the DNA methylation actually in the cultivated, we lost the DNA methylation. So when we lost the DNA methylation, we have the degradation of the polyphenols. And these two genes, peroxidase and the beta-galactosidase, these two, what they do is they degrade the polyphenols. So this is methylated in the wild type. It's not methylated in the cultivars. So what happening is we have less polyphenols in cultivated olive and more polyphenol, which is a very important thing. The most expensive oils are the oils high in polyphenol. This is really good for using DNA methylation for breeding programs. Second one, the lipoxygenase right here is a gene that takes the linoleic acid and make volatile compound out of it. Volatile compound, when you smell the olive oil, that smell, it's coming from the volatile compounds. And this gene right here is methylated in the cultivar, but it's not methylated in the wild. That's why the olive oils from wild types are grassy. You smell that grass cut, fresh grass smell, but it's not in the cultivated. So this is for people breeding for flavor and smell, because it's very important. Les qualités sensorielles, they can use this as a biomarker. The last one is the FAT3. So we're talking about the FAT2 and the FAT3. So this in the olive, the FAT3 is actually controlled by methylation. And this is going to be the subject of our next publication. Here is just again we took those 4 genes, we compared them between the wild and the cultivated. We found out that the cis element are the same. Here it's the same. So if we have the same everything and we have 2 different phenotypes, it's most likely due to the DNA methylation. For the other genes, the same things. For the peroxidase, the same things. For the beta-galactosidase, the same thing. So anything related to polyphenols in olives, they are due to the DNA methylation. The last one, we found some differences when we compared the wild to the cultivated, which is the fatty acid desaturase. So here, the pressure of selection affected the promoters of the wild and cultivated FAT3. That's we have a different concentration of oleic acid and linoleic acid between wild and cultivated. This is just to summarize the whole thing. If you look at this, you will understand the entire analysis that we did. And I think that's it. Thank you so much.
Thank you very much, Usama. That was a great presentation. I enjoyed it very much. Now we will open it to questions. If anybody has some questions for Usama, you can unmute yourself and ask your question.
Okay. Thank you, Usama, for the presentation. It was wonderful. And about the approaches. You talked about approaches. It was 3. You talked about 3 approaches.
Yes.
Yeah. And I need more clarification.
So to annotate a gene or a genome, you need to make sure what you're talking about is true. So when you combine 3 different approaches, you're trying to convince the reviewer that your data and your results are legit. So when you combine those 3 approaches, the first one is based on open reading frame. It's a de novo annotation. You have your algorithm is basically walking your DNA base by base, looking for an open reading frame. Les ORF. Les ORF très important because they will tell you where the gene is. And the annotation is about genes. I want to find all the genes that are in my genome. So when the algorithm will find an open reading frame, it will start from there. You start from there, you're looking for the longest gene. You're trying to stay away from stop codon as much as you can. These are fundamentals. We all know that. You find the open reading frame, your DNA polymerase is trying to give you the longest gene. That's the first approach. The second approach is by similarity. You take that sequence of DNA and you align it against a database. The database has different organisms, those organisms, they have genes that are really close in sequence. Sometimes you have the single copy orthologs. Single copy orthologs are genes that are in every single plant species. They're all the same sequence, they do the same thing. That's your base. That's the second approach. The third approach is an RNA Seek approach. RNA Seek, what you're doing, you're only sequencing the coding part, which is the messenger RNA. That's it. So how many transcripts you have there? If you take that and you compare it to what you found from the different approaches, it will confirm it. Like we said here, when we used the RNA approach, we only confirmed 93% of all the genes. We start with 50,000, but then we used the RNA seq data and it confirmed only 47,124. That's why you combine the three approaches. So you are almost, when I say you're right, but you're as close to the reality as possible. But then when you do the function annotation of those genes, even that number will drop down because some of those genes, they are not coding. And if they don't code, they don't have a hit on the BLAST database, they're most likely to be a non-coding gene, like a transposable element or retrotransposons or something like that. So that's why the necessity of combining the three approaches.
Thank you.
Good. Any other questions?
So, you studied the wild olive trees and domesticated. So did you study all the variety, like the European or the Abiquina or the Moroccan one? So
well,
what was?
Yeah. Go ahead.
Oh, it's just I want to know what kind of which one did you use in your study?
Okay, the one we used in our study was they were from Turkey.
Okay.
Because the project was supposed to be a big project, and then it turned to a small project, and we couldn't sequence a big number of trees. But why we took the ones from Turkey, because the cultivar and the wild type from that part of the Mediterranean, you cannot even separate them. Okay. Genetically, you cannot separate them. They're very tight. So we're looking for something that is so tight that the only way we can find any differences, it's going to be on the regulation. And the study that was published in 2019, they took Spanish varieties, Italian, French varieties, all the Mediterranean, and they sequenced the RNA to dig more deep into the DNA, and they couldn't separate the gene expression profiles. That's how, especially in the Eastern accessions, which is anything coming from Turkey, from Syria, Lebanon, or Palestine. So those were so tight. That's why we sequenced that. And that was the reference genome that we sequenced before. The wild one, the reference genome, was exactly the same one we resequenced for this purpose. But what I would love to do, if we can have a project, I want to, you know how diversity goes east-west, but not north-south? If we can take the varieties, especially, this is to protect ourselves, because the French government took a lot of clones and now they call them La Picholine Française or La Picholine de Languedoc or whatever. To protect ourselves, we take the southern coast, we and we compare it to the northern coast, because east-west, we know what happened. We want to know if in the history of domestication, if anything was stolen in the hundreds of years of colonization.
Yeah.
That will tell you that whatever they're talking about is true or not. And here's another thing. The varieties from Morocco, Tunisia, Algeria, especially the ones from Morocco, subhanAllah, they have more oleic acid than all those Spanish varieties, and they have more polyphenols than any other of the other varieties. And there is a doctor here in the United States, in Los Angeles, who goes to Sus, and he gets olive oil from wild trees, and he's selling it here as medicine, high polyphenol, $45, 350 milliliters.
Yeah.
This is death.
Dr. Grandy, right?
Gundry, yeah.
Yeah, Gundry. Yeah, I saw that.
كيعطيك شمة ب$50. This is we need to have a national program. We need to have.
I know.
انت خاص تكون عندنا une volonté nationale, royale, call it whatever you want. We need to protect those species. الأرجان, le carobier. I don't understand why till now المغرب ما بغاش يدير بروجي على الخروب. كاع الاندستري اغرواليمونتير. Xanthum anytime.
I agree with you completely. They made a big mistake about argan tree. They should have patented. Now it's they growing it in Israel.
Even
about the olive trees, there is a difference between north and south. I did my study in Spain, and even in Spain, between South Spain and North Spain, north of Jaén and south of Jaén, it's a different kind of trees, and they give different quality of olive oil. And in Morocco, between the Atlas and the east of Morocco and north of Morocco, for example, the region, the most the best that has the most rich of polyphenol and yes, in the region of north between Rafsay and Fes, and between Shawan and Wazan. They have very high and very high productive olive. The normal olive tree that give you around between 17 and 8 and 22 liter per 100 kilogram of olives. In Wazan, it give you up to 33, 35 liter per 100 kilo of olives. It's crazy. There is old trees. But going back to, we need to patent the variety of trees that we have in Morocco, because in Spain they are in the region of Valencia. They are producing the same oranges that we have in Morocco, Navel, and they are now fighting Morocco that's selling them to the European Union to pay them a royalty for having those trees in Morocco. The thing that you're saying, it's a big project, and I think we need to act yesterday, not today.
There is only one way to. I'm going to tell you this. We need a genetic variability assessment of every genotype of olive, argan, carobier, and cannabis. Cannabis is next. I'm going to tell you this. I have companies from here that we met in San Diego, from America, from Israel. They want to put their hand on some genotypes from Morocco. Why? America is coming out of, they're legalizing cannabis in America. So what's going to happen, the production going to come out of the greenhouses to the land. So they are looking for genotypes that are resistant to disease, they're tolerant to drought, to cold. And if you look at the northern region of Morocco, from the coast to the mountain, the gradient that we have in there will allow to have different genotypes that will be adapted to different ecosystems. People are coming for those people. If you don't want to do it legally, they're going to pay someone to give them the seeds. They're going to sequence that, put a patent on it. We need people to push for une volonté nationale to tackle these 4 species. We will be making billions of dollars if we take care of those 4 species. That's the future of agriculture.
Well, that's true. We need now to find, now that we are talking about this, we need to write it, make like a good dossier and take it to the Minister of Agriculture. And I think this is if there is like a will, we can do it. And we need, now that we know about this, now that we are outside Morocco, we can help the people and look at our contacts and try to do it, because the money is there. And with the new there is people, they have good intention in Morocco, they will help us to do it. But we need to prepare for them dossier, how much it's gonna cost to do all the sequencing, you know, and
now
everything.
It doesn't cost much to sequence. Now we're talking about a couple $100 per sample.
But
it's really
the most expensive, it's the legal stuff, to patent it and to register it, so nobody use them. And if anybody use them, we can like go after them and they will pay for it. And you know, Monsanto, if they put their hand on this, it's gone.
Well, now you don't have to worry about Monsanto because Bayer bought them. They bought them for $40 billion. So no more Monsanto. Bye-bye.
Yeah, okay, good. So we need to wrap it up. I have 1 or 2 questions, Osama, about the, of course, when you talk about methylation, we have also demethylation, switch on and off. So I want to know, during your study, did you find demethylases within the genome of olive oil? And if yes, where these genes are and where these demethylases are activated or expressed?
So we mostly studied the methylation instead of the demethylation, but I found a total of 43 demethylases in olive genome. 17 of them were downregulated and the rest were upregulated. And that's what took actually the methylation down during the ripening of the fruit. This is what we were talking about. That's why is it going up in the leaves, but it's going down in the fruits? You have 2 answers. It's either the methyltransferases in the fruit are not upregulated, and on top of that, you have demethylases. We're going to go back to that right here. Right here. See here, this one. This is the fruit. This is the methyltransferases of the fruit. If you compare July to November, it's the same. There is no big difference. Not like here. Here you can see the difference. So this is just a heat map. We're giving you an idea about the expression of those methyltransferases. So, but then we go to the demethylase right here, and you look at the demethylases in November are slightly, if you compare this shade of color to this one, these demethylases are highly expressed in November compared to, so that's why we have a hypomethylation with the ripening of the fruit.
Okay, thank you very much. I really appreciate all of you who were here, and especially our presenter, Osama. And of course, I will put this on YouTube for people who could not make it today. Thank you again, everyone, and Assalamu alaikum.
Wa alaikum salam wa rahmatullahi wa barakatuh.
Thank you. It's good study.
Yeah.
Congratulations.
Thank you.
Islam.